Hello, bio people. Today we are going to talk about non-Mendelian inheritance. So in last lesson, lesson 1, we talked about Mendel and his work with garden peas and his studies and his scientific method and his conclusions that some traits are dominant and that traits are assorted or separated independently of one another were concluded based on his massive amounts of data. Today we're going to talk about a couple of types of inheritance that don't fall under his exact rules. The first one is incomplete dominance. Homozygous parents have offspring that show an intermediate trait, so something in between the parents. And homozygous parents, you'll remember, have the same trait, the same two alleles. So whatever the alleles are, they're the same. Neither trait, though, is considered dominant or recessive since one does not conceal the other. The heterozygote shows an intermediate blended phenotype of the two parents. So an example would be these flowers. You have a red flower and a white flower, and when you cross them, you get a pink flower. You get the pink flower because the alleles for pigment are only at 50% because half of them are red and half of them are white. And so you only get 50% of the red color in the flower, making it pink. You do not get any red or white visible individually. When we look at a cross like this, we start out with true breeding flowers. So these are true breeding red flowers. So they would be two alleles that are the same. And the white flowers have two alleles that are the same as well. And when they cross, we get 100% pink flowers for our F1 generation. We let these guys self-pollinate, and then we wind up with a ratio of 25% red, 50% pink, and 25% white. This ratio is the same ratio we would get with a normal monohybrid cross, but it would be our genotypic ratio. In a monohybrid cross, your phenotypic ratio, or what you see, the trait you see, is a 3-to-1 ratio for a monohybrid cross. So this is not following Mendel's normal ratio combinations. There are a couple of rules for incomplete dominance crosses just to keep things organized and easy to read. We use capital letters to represent the alleles because neither trait is concealed by the other. Usually we use the first letter of the phenotype. So the phenotype is what you see, the physical trait. So for an example, red flower would be 2 dominant R's, white flower would be 2 dominant W's, and the pink flower would be 1 of each. So this is we have a couple of practice problems for you, and I'm going to do the first one with you, and the second and third one I'm going to walk you part of the way through, and you're going to finish it on the worksheet. So if you look at this cross, we are crossing a red flower with a pink flower. When you do that, you bring the letters down into the box. So you bring it into this Punnett square. You bring down and you bring across. And you bring down and you bring across. Now, because you're working with 2 different letters, we sort of follow the same rules that we do with a monohybrid cross, where you always put the dominant letter first and then the recessive letter, like you're spelling a name. But here, we're going to keep the R's on the left and the W's on the right, unless, of course, we have 2 W's. But we want to keep it consistent so when you're looking at it, you understand where the blended ones are. So if we have done our Punnett square, and the question we're asking is, what percentage of the offspring may be red and what percentage may be white? And we're going to emphasize the fact that we're saying may be here, because what does a Punnett square tell you? It tells you the possible outcomes that are available. If these are the possible outcomes, but you don't have 4 individuals, you don't have 4 offspring, you might not get one of each. You have, what it is for each individual, for each offspring, there's a chance, a 25% chance plus a 25% chance. So here, a 50% chance that they'll be red and a 50% chance that they'll be white. If these 4 boxes each had a different combination in them, then we would say there was a 25% chance they would be red, a 25% chance they'd be white, a 25% chance they'd be pink. Did I say they were white before? I meant pink. So you know that each box is worth 25%, and then obviously if they're the same, you add those percentages. So here we have, for the red, we have a 50% chance that it would be red and a 0% chance that it would be white, because none of these squares have a white genotype. So for the next one, I want you to bring your letters down and across. Remember, keep the R on the right, on the left, and the W on the right. Fill that in, and I'll give you a minute. All right, so now that you've filled this in, the question you're asked is, what percentage of the flowers may be white? So now look at your squares. Remember, each square is worth 25%. So add your squares up, and that will tell us the percent that will be white. Put that into your note taker, please. A third practice problem, how many phenotypes would you see in the offspring? So this one's got the question up front for you. So you're going to bring your letters in. So bring them down and then across. And looking at your 4 squares, you're going to determine how many different phenotypes. And for this, again, we have the phenotype choices are red, white, and pink. So in this box, you will write the percent red, the percent white, and the percent pink. All right, so that's an incomplete dominant cross. So you're going to get something that's blended, something that's in between what you started with. For dihybrid crosses, we're looking at 2 different traits at the same time, and we're looking at traits that have a dominant and a recessive option. So slightly different from what we just finished. We talked a little bit about this in the first lesson, but we're going to kind of dive deep in this lesson. So we're going to work with F.O.I.L. Just remember that a dihybrid, this is 2 hybrids. So a dihybrid cross is looking at crossing this individual with this individual, and each of these individuals is a hybrid. Okay, they are each hybrid or heterozygous for both of these traits. This doesn't work the same way anyway. This doesn't work out if you use an individual that's not heterozygous or hybrid for both of these traits. So if you were crossing someone who was homozygous dominant for color and heterozygous for trait, you're not going to get the same Punnett square workout. So looking at these, we want to make sure in our monohybrid crosses, it was pretty easy to figure out where the alleles for the gametes would go on our square, because you only have 4 boxes. Forgive my crooked line. Okay. You only have 4 boxes, and you have 2 alleles for each individual, right? So if we were talking about height, and we had a heterozygote here, we would put one of the alleles here, one of the alleles here, and then if we were crossing it with a tall individual, we would put the two alleles here. Then we would bring them into the boxes, and we would see that we have 100% tall, 50% of that is heterozygous and can pass on that short trait later, but at this point it's not. So we want to make sure that when we set up the gametes for the dihybrid cross that we have all the combinations. So these are representing the gametes and the allele the gamete would be carrying. So these would be your eggs and these would be your sperm. And they're each carrying one allele because they're taking one allele to fertilization, and they each add one and you get a total of 2. So here we'd use F.O.I.L to figure out what we're going to do with these 4 different alleles and how we can combine them into the gametes. So your first allele is this one. The outside are these 2. The inside are these 2. And the last are, the last are these 2. So looking again, first outside, and then inside and last. Okay, so inside and last. For this parent, that means that for the first, I have a dominant Y and a dominant R. For the outside, I have a dominant R and a dominant Y and a recessive R. For the next one, I have a recessive Y and a dominant R. And then for the last, I have 2 recessives, the Y and the R. Because we have the same exact genotypes for both of the parents, we can repeat the process over here and we get the same gametes. Okay, so these gametes would be sperm, and these gametes are eggs. Why are the 2 sets of possible gametes the same? Because we start with the same genetics, the same genotype. Okay, so we have the gametes. Now what do we do? Because we have 4 possible allele combinations in our gametes, we need a bigger square. And since we need to get the same possible combinations in each parent, we can actually put them in the same order on the Punnett square to make it a little simpler for us to follow. So that's what we do. We take a 16-box Punnett square, and we put our gametes across the top for the female. So these are the 4 gametes we decided on in the other slide. Here's the gametes for the sperm. And then we cross them. So you're going to cross all 16 boxes, so you bring down the letters from the top, the letters from the side. So the alleles coming down here are a dominant Y and a dominant R, and the alleles coming over are also a dominant Y and a dominant R. A couple of rules for this Punnett square. We want to keep our letters together, so we want to keep the 2 Y's together, and we want to keep the 2 R's together in each of our squares. We also want to keep the same 2 in the front. So if we start out with the yellow in the front, we want to keep, the color in the front, we want to keep the color in the front and the shape next to it. That way, when you're reading it, you're less likely to make an error. So when we read these are dominant, so yellow and round, dominant, but we have a recessive here. But this recessive doesn't matter because with one dominant, you're going to be round. So when you are looking at these, you need to look to see if you have at least one dominant trait, you are going to have that dominant phenotype. If you have a recessive allele, you have to make sure it's not a two recessive allele. So here we have a heterozygote match. So this means it's going to be dominant and show the dominant round phenotype. However, here we have two recessives. So this P is going to be wrinkled because the recessive trait is wrinkled. So when you're looking, if you've got at least one dominant Y and one dominant R, the P is going to be yellow and round. So that includes 1, 2, 3, 4, 5, 6, 7, 8, 9 of these. So this is the 9 yellow round. Then we have 3 that are going to show one recessive trait and one dominant trait. So the green here is the recessive and the round is the dominant trait. So to find green ones, we have to have 2 recessive Ys and at least one dominant R. So that includes these 3 right here. For the yellow wrinkled, we look for the yellow is dominant, so we need to have at least one dominant yellow. The other allele doesn't make any difference at this point. And then we want it to be wrinkled, so it has to have 2 recessive R's to show wrinkled. And then down here, the recessive, they all have to be recessive alleles to show the green and the wrinkled. So if you're looking through the box, you look for a dominant Y with 2 lowercase r's, or 2 recessive R's. You have this one, and this one. And there's only one double dominant with the green and the wrinkled alleles. So every time you set up one of these boxes, you want to be aware that you should be finding a 9:3 to 3:1 phenotypic ratio, and phenotype is what they look like. Their genotypic ratio, you can see from all these letters, is a lot more complicated than 9:3 to 3:1. If you want to double check your math when you're counting up your squares, your math should equal 16 boxes. So 9 plus 3 plus 3 plus 1 equals 16. After observing these dihybrid crosses, Mendel came up with the law of independent assortment. And that means that the genes for a trait are going to separate into gametes independently of one another. So the genes for traits for the color and the shape are not on the same chromosome, and so they are not going to affect each other as far as when we get a dominant or when we get a recessive allele, because they assort independently. So non-homologous chromosomes align independently during meiosis in metaphase 1. The classes of gametes are produced in equal amounts, so you got one of each in every gamete. And these are for this particular cross we just did, those are our choices. This is only true for the genes that are on separate chromosomes or on the same chromosome, but so far apart on that chromosome that we have crossing over happening very frequently on that chromosome. So remember back from the meiosis chapter, we talked about crossing over where the tetrad of chromatids work together and they actually break and reform and swap alleles for the same traits. So it's not a mutation because they're not changing, they're not harming the gene, they are just swapping the choices for that trait. Okay. The independent assortment is basically determined during metaphase 1, how these guys line up. Remember, all of this information that we're talking about, Mendel was figuring all of this out without any knowledge of DNA or genes or chromosomes. He didn't know any of that stuff. All he was doing was looking at his phenotypes and backing it up to figure out what the genotypes were. So he knew the traits were controlled differently, but he didn't understand that they were alleles for the same gene that he was working with. Okay, so let's review Mendel's laws real quick. We have the law of segregation. We can see that in a monohybrid cross. We're looking at a single trait, and each allele segregates or separates into different gametes so that you only have one allele per gene in each gamete. If you wind up with extra alleles, you're going to have too many, and that will cause problems in the offspring. The law of independent assortment happens for dihybrid crosses, so you're looking at more than one trait, and it's basically saying that those genes are going to separate or move through the process independently of one another. They are not on the same chromosome, and so therefore during meiosis they will move in metaphase, line up differently without care of what the other one is doing. And the law of dominance, again, is that one trait would be able to conceal the presence of another trait in the phenotype. So, if you have any questions, please bring them to class so that we can talk about them. Thanks for listening.